Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your thin layer chromatography separation of simple lipids. Okay, so this what does this word means chromatography? See, chromatography is simply means that separation of component by using stationary phase and mobile phase. Okay. So in chromatography, basically, what should we do? We separate the component into we separate the component in component by using the stationary phase and the mobile phase. Okay. In thin layer chromatography, there are again two phase. One is your stationary and one is your mobile. Okay. In thin layer chromatography, this stationary phase is a glass slide. Okay, and mobile phase is what? Mobile phase is or solvent. Okay, and in glass slide there is a layer of silica gel are introduced. Okay, what should we do? We make a paste of silica gel. Suppose that this is your slide and here it is the silica gel paste. Silica gel paste. Now we pour this silica gel into the glass slide. Okay, suppose that this is your silica gel. Okay, when it will dry, we make a line 1 centimeter above the slide and 1 centimeter lower the slide. Okay, and here what should we do? We put a sample. Okay, we put a sample and kept this slide into a solvent. This is what? This is your solvent. Now, due to this capillary action, this solvent move to the upward direction, and when they move the upward direction, the component react with the solvent or make a bond with this solvent and move to the upward direction. Suppose that there is a component in which B and C are present. Now suppose that B have a more affinity to the solvent. So what happened? They move faster rate and reaches here, and C have less affinity to the solvent, so they move lower than the B. Okay, now this is about the thin layer chromatography. Okay, now come to the next slide. Here the objective of the experiment. What is the objective of this experiment? We have to separate the lipids present into a food product by using the thin layer chromatography. Okay, and after separation we have to identify the lipid. Okay, so again we using the TLC. So this is the diagram for it that I already told you in the first slide. Okay, so what should we do? First of all, we have to separate this lipid, and after separation, we have to identify the lipid by using this thin layer chromatography. So here, when we separate the lipids, means fat, these fats are soluble in organic solvent. Soluble in organic solvent. So what should we do? Either we use ethanol, okay, or either we use hexane, depend upon the types of the lipid that we want to separate. Okay, so we take a solvent and kept this slide into this solvent and wait for a day, and due to this capillary action, this lipids present into a food products are separated. Okay, now the next one is your principle. See, TLC is a separation technique that allow for the distribution of compound between a mobile phase and a stationary phase. That I already told you. This TLC is what? TLC is a separation technique. Okay, they are what? They are a separation technique which separate the compound between the mobile phase and the stationary phase. And most classes of lipids can be separated from each other by adsorption of chromatography on thin layer. Okay, now in TLC a thin layer of stationary phase is bound to a inert support. ठीक है, that is your glass plate, plastic or aluminum sheet. That I already told you here. What we have a slide. Okay, this is a slide of glass. Okay, and we have a stationary phase, that is your silica gel. Okay, this silica gel is what? These are bound to an inert support, and this time the inert support is what? It is a glass plate. The sample and the standards are applied as a spot near one end of the plate. That I already told you. We make a line of 1 centimeter just above the slide end, and here we put the sample. Okay, now this sample is this plate is placed in a developing chamber with the end of the plate nearest the spot being placed in the mobile phase. This end in which we put the sample, it should be placed near the mobile phase at the bottom of the chamber. The mobile phase migrate up the plate by capillary action. Now due to this capillary action, this mobile phase goes upward, carrying and separating the sample component. The separated bands can be visualized or detected and compared to the separation of standard compound. Now what should we do? We separate the separated band and visualize or detect it by comparing the separation with the standard compound. Now the next one, that is your chemical used in this experiment. First, we use a acetic acid. Case number 64-19-7, and the hazard related to this chemical is what? They are corrosive in nature. Now the next is your diethyl ether, that is your 60-9-7, and the hazard related to ethyl ether is what? They are harmful, extremely flammable. And the next one is your hexane 110-54-3. What? They are harmful, they are highly flammable, and even dangerous for the environment. What does this word harmful means? See, harmful is related to the animals, is related to the human beings. Okay, so if human beings come in contact with this chemical, then it will harm the health of the human being, or either if the animals come in contact with this chemical, they will also harm the animal. Now the next one is your sulfuric acid, that is 766-4-93-9. They are what? They are corrosive in nature. So these are the list of the chemical that we use in our experiment, that is your acetic acid, diethyl ether, hexane and sulfuric acid. Now the next one is your reagent used in this experiment. First of all, the reagent first we use in this experiment are what? They are a chloroform and methanol. Okay, and the ratio of chloroform and methanol is 2:1. That means that if we take chloroform 2 ml, then we have to take a methanol 1 ml. Okay, that is 2:1. Okay, and the mobile phase will be a hexane or the diethyl ether or acetic acid in the ratio of 78-20 or 2. That means that we take a hexane, mixture of hexane, diethyl ether or acetic acid. So hexane will be 78 ml. Okay, and this diethyl ether acid, the diethyl ether will be 20 ml. If this is 78%, this is 20%, and this is 2%. So this acetic acid will be 2 ml. Okay, and we also have a standard of triglycerol, fatty acid, cholesterol, ester and cholesterol. Okay, we also have a solution of concentrated sulfuric acid in 50% of aqueous solution. So these are the list of the reagent that we use when we performing this chemical. Now the next one is your hazard precaution and waste disposable. We all know that we are using two acid. One is your sulfuric acid and one is your acetic acid. So these acids should be used in a fume hood, and we all know that this dry ethyl ether is what they are flammable in nature. So we have to um, avoid it to open it around the flame. And we also have to follow the other normal laboratory safety procedure. We have to wear a gloves, we have to wear a glasses, we have to wear a lab coat, we have to wear a proper shoes. And one more thing that is very important when we performing the experiment in the lab, we have to avoid eat anything inside the lab. And this diethyl ether and hexane are what they are hazardous waste. So when we are handling it, we have to take a extra care, and the waste produced by these chemicals should be discarded in a hazardous waste, and other waste should be drained out with water rinse. And along with it, we have also followed the good laboratory practices which are outlined by our environmental health, and we have also followed the protocols which are given by our institution. Okay, now see what is written in the slide. Use acetic acid and sulfuric acid in a fume hood. Diethyl ether is extremely flammable, is hygroscopic, and may form explosive peroxide. Otherwise, adhere to a normal laboratory procedure. Wear safety glasses at all time, and diethyl ether and hexene must be handled as a hazardous waste. Other wastes like may be put down the drain using a water rinse, but follow a good laboratory practices outlined by our environmental health and safety protocol at your institution. Okay, so the next one is your material used in this experiment. The first material that we required is a capillary tube or syringe, and this is required so that we apply the sample into a plate. And we also required a developing tank along with a lid, filter paper, Wattman number 1 to line developing tank, oil fat food sample, that is either we required a hamburger safflower, prepare at a concentration of 20 milligram per ml, that is 2:1 chloroform methanol solution. So what we have to do, we take a sample and add this chloroform and methanol solution in the ratio of 20 is to 2 is to 1. Okay, and the amount of the sample will be 20 milligram. So in every 20 milligram of sample, we have a 2:1 chloroform methanol solution per ml. Okay, so what we have to do, first of all we have to prepare this chloroform and methanol solution. Okay, and it will be a 2:1. Then from it we take 1 ml, and this 1 ml uh, will be used to dissolve the 20 milligram of food sample. Okay, so along with it we also required a pencil, thin layer chromatography plates. Okay, and this thin layer chromatography plates uh, will be poured with this silica gel 60s, and the thickness will be 0 to 0.25 mm, which are coated backing 20 into 20 centimeter em size. Okay, now the next one is your equipment required for this experiment. We required a blower, that is blow a hair dryer or oven. Okay, so these two equipment we required, one is your air blower and one is your oven. Now the next one is your procedure. First of all, what we have to do, we have to prepare a silica gel plate. For this first of all we have to dry the plate by putting it into a oven at 110 degrees Celsius for 15 minutes. Then after that we have to cool it at a room temperature for a 5 minutes. Then we draw a, suppose that this is our plate. Okay, Now what we have to do, first of all, we dry it at 110 degrees Celsius for 15 minutes. Okay, after drying, we cool it for a 5 minute. We cool it for a 5 minute. Okay, then after that, we draw a mark of 2.5 centimeter. Okay, and then again we make a mark with a pencil which divide the plate into a 10 lines of equal width. Now uh, with the help of this capillary tube or syringe, we apply the 10 microliter of standard and sample to separate the line, and application should be done as a streak below the original line. Write the identity of the sample standard in each line. Allow the spot to dry. You may accelerate drying by using a low temperature air blower and write your name in the top right corner of the plate. See, first of all, what you have to do, you have to dry this. Okay, after drying, you have to pour the silica gel into it. Okay, after pouring the silica gel, you have to again kept at a oven at 110 degrees Celsius for a 15 minutes. Then again you have to cool down. Then from the bottom you have to make a line of make a one line above from the end of the plate 1 centimeter, and here you have to put a sample that is 10 microliter, and then you have to um, put this plate inside a solvent. Okay, now see what is written in the slide. Place a plate in oven at 110 degrees Celsius for 15 minutes, then cool it to a ambient temperature for a 5 minute. Now with a pencil, draw a line to mark the origin 2.5 centimeter from bottom of the plate, and make a mark with pencil to divide the plate into a 10 line of equal width. Okay, suppose that here what should we do? We make a line of 10, and it should be a equal width. Now use capillary tube or a syringe to apply approximately 10 microliter of each standard and sample to separate line. Use a middle 8 lanes. An application should be done as a streak across the center of the lane origin. This is the best accomplished with your four spot of 2.5 microliter of each. Now below the origin line, write the identity of the sample, which means that we have to write down the name of the sample, and then allow the spot to dry, and you may accelerate drying by using a low temperature air blower. Okay, if we want to accelerate the drying, we uses the air blower, but the temperature of the air blower should be low. Now write your name in the top right corner of the plate. Now the next one is your developing of the plate. Line the developing tank with the Wattman number 1 or similar filter paper, standard distance from origin RF value. Now pour the mobile phase gently over the filter paper until the depth of the solvent in the tank approximately 0.5 centimeter, about 200 ml is required. Then place the lid on the tank and allow 15 minutes for atmosphere in the tank to become saturated with the solvent vapor, triglycerol, fatty acid, ester, cholesterol, sample spot number. Now place the spotted TLC plate in developing tank and allow it to develop until the solvent from reaches 0.2 centimeter from top of the plate. Now distance from origin RF value is identified. Okay, RF value means that suppose that this is your plate and this is your line. Then then a component travel here from here, so this distance are called what? Are called the RF value. Now remove the plate from the tank and immediately mark the position of the solvent front. Evaporate the solvent in the fume hood. Okay, now what should we do after um after removing the plate from the solvent? Then we mark it to mark it, and after that we evaporate the solvent into the fume. Now the next one is your visualization of the lipid. How we visualize it? For the visualization, in a well ventilated fume head, we spray a 50% of aqueous sulfuric acid. Okay, now after that we have to allow it to cool down. Then we heat the plate for 5 to 10 minutes at 100 to 120 degrees Celsius. Remove from the oven, cool and inspect. Handle the plate with caution as the surface still contains sulfuric acid. Now mark all the visible spot at their center and note the color of the spot. Okay, so what should we do? We place it into a fume hood and spray 50% of aqueous sulfuric acid and put it into for a drying. Then after that we heat the plate at 120 degrees Celsius or 100 to 120 degrees Celsius in an oven for 5 to 10 minutes. Then remove it and cool it and inspect it. Okay, so when we handling this plate, we have to take care because it is still contain a sulfuric acid. Now after that we mark all the visible spot at their center and also note down the color of the spots. Okay, data and calculation. For the spot, each of the standard and sample report the distance from the origin for the spot. Also for each spot we have to calculate the RF value that I already told you. Suppose that this is your spot and this spot along with solvent move here. So this distance is called what? It is called a RF value. Okay, as the distance from the origin to the spot divided by the distance from the origin of the solvent front. Using the RF value of the standard, identify as many of the spot in the sample as possible. Now this is the table of the experiment. This is the standard in which triglycerol, fatty acid, cholesterol, ester and cholesterol. Then here the distance traveled by the origin and this is your RF value. Now this is the sample spot number, distance from the origin, and with the help of this we find out the RF value and identity. Okay, so along with it, you can also write down here the oil or fat type which are tested, like your either it is tested triglycerol or fatty acid or cholesterol ester or cholesterol. So this is all about the determination of lipid with the help of TLC, that is thin liquid, that is your thin liquid chromatography. I hope you will understand. If you have any query, just let us know in a comment box. And for more detail, you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and you can also subscribe ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe. If you have any query, just let us know in a comment box.